Hi, today I have come to show you the results of the ubiquity of microorganisms. So I did the stroking of the plates or infecting the plates with the environmental sample in TSA, SDA and blood agar plate. So I'm going to show you how it appears. Maybe you'll find it interesting. So this is from my fridge. As you can see that this is a TSA plate, and you see that the labels, one more time I'm showing you, they're at the edge. So this part you can see, this is actually from the swab from the fridge, and this part is from my shoes. So we can see how they have grown over here. The type of growth is also different. Maybe the shoes one will develop a mold, I'm expecting. Let us wait and see. But this is the growth of a swab, one from the fridge and one from the shoes, and this is on TSA plate. So this is another TSA plate, and the swab was taken from the soil. You can see this portion, and this part, the swab was taken from my skin. I have done this to find out if on the skin Staphylococcus epidermidis is there. So I will actually culture it later and I'll find it out. And definitely Staphylococcus epidermidis should be there. I'm expecting that. Anyway, so we are done with this result. Let me put it there. Now this is the SDA plate. So this one also from shoes, and this part is from the fridge. But the thing is that it seems my shoes are more clear, more clean than the fridge. As you see, less number of colonies over here, and from the fridge is a huge amount of microbes are growing. And this is SDO plate. Always remember that you see the writings are on the edge, not over here or not on the top. This is written by the brave staff over here, but we should not write anything on the top so that we can see the growth of the colonies clearly. Okay, so now the most interesting one comes over here. This is the blood agar plate. So in this blood agar plate, I actually chose three portions of my body. One is the feet, I took the swab. Another is the nair, and another is my fingers. So let me show you. You can see that especially on the feet, as I'm holding, you can see that light is passing through. This is showing the beta analysis, beta hemolysis. Can you see that? Let me show you. How can you see that light is passing through? Okay, is it clear? I don't know. Okay, maybe this is good. Okay, now if you clearly look over, if I can show you, some of them are greenish colonies, like 1, 2, 3. Those are greenish colonies. Those are actually alpha hemolysis. That means partial breakdown of the blood or hemoglobin has been done by the microbes, in comparison to the beta hemolysis, which is done by the microbes who actually did the complete hemolysis of the blood cells. Now there are more of the colonies over here, we can see at the top, but those are not making any green color because you cannot see from back. They are not making any greenish color, so they are making the gamma hemolysis. That means they are growing over there, but they are not making they are not breaking down the blood cells. So that's it. But I want to show you one more time that we always keep the plates inverted. The reason is that, as I told you in my earlier video, that when they are kept in incubator, then they are in a little bit warmer temperature, 37 degrees centigrade. Then we actually put them in the fridge, we stored them, and there by condensation the droplets of water will fall. If it is like this, and that will fall on the colonies and the colonies will be messed up. So we always keep the agar plates in inverted position. Now one more time, 1 point more. Have you seen the blood agar plate and this one where the human sample is? I just sealed. I didn't have the parafilm, so I sealed with the sellotape I have. So I sealed this because this is human sample. We have to do that always. Seal the plate all around. Okay. So that's it for today.